Welcome everyone to our online NPTEL course Environmental Chemistry and Microbiology. This course will be taught by Professor Sudha Goel and myself Professor Anjali Pal. We are from the Civil Engineering Department IIT Kharagpur. We have divided this course into two parts. The first part is environmental chemistry, that will be covered by me, and the second part is environmental microbiology, that will be taught by Professor Sudha Goel. So, in my first module I have discussed about the acids, bases and salts. In the second module I discussed about the chemical equilibrium. In the third module I have elaborated the chemical kinetics. In the fourth module I have discussed about the catalysts and catalysis, and this is my fifth module and lecture number 22, where I am discussing about the nitrogen chemistry. This is the part B. In my last lecture I have covered the nitrogen cycle and nitrogen chemistry, that was part A, this is part B. The nitrogen data, why it is so important? In this lecture content I will tell about the significance of nitrogen data. I will tell how we can determine the ammonia nitrogen. I will also discuss about the how to determine the organic nitrogen and the nitrite and nitrate nitrogen. So, importance of nitrogen data as an indicator of sanitary quality. This is a very important figure. It is known, it was known for long time that polluted water would purify themselves with time. So, if you spend enough time, and this is of course the natural pollution, say for example, you put some organic matter which is biodegradable organic matter, say for example somewhere, okay, then say for example in some natural environment you throw it, then what will happen? Naturally it will be purified with time. Okay. The possibility of health hazard with this water would decrease with time. With time the hazard will decrease and temperature. So, if you can see here the effect of temperature, you can see in the winter season and summer season. In the winter season we know that temperature is lower compared to summer, and this is the health hazard, and then you can see that with time the health hazard is decreasing. So, water is becoming purified, okay. But in summer you can see it is going faster, okay. You have seen the kinetics. So, this is the kinetics. So, here it is a time. So, its temperature also has some effect, and also the time, okay. So, this is a natural purification, okay. And if you can measure different types of nitrogen, in my previous lecture I have already told that nitrogen is present as ammonia, nitrogen may be present as organic matter, nitrogen may be present as nitrite, it may be present as nitrate, okay. So, there are different forms of nitrogen. Now, if you can measure these different types of nitrogen, how much is present in the water, say for example, pollution starts here, and then you can see with time how the nature of combined form of nitrogen is changing. If you measure the organic nitrogen, then you will see that with time, this is the time, you will see that organic nitrogen concentration is decreasing. This is the nitrogen in milligram per liter. So, this is decreasing, organic nitrogen is decreasing, okay, with time. And you will see that ammonia nitrogen, the concentration is increasing, and then it is increasing and it will go to a peak value, and then it will again decrease. Now, in between you will see that nitrite nitrogen, this is increasing, and then finally it is decreasing, and nitrate nitrogen, it is increasing with time. Now, why it is so? Because Say for example, you have polluted with some protein molecule, okay. Then what will happen with time? The protein molecule will be first decomposed and it will form the ammonia. First it will form the ammonia that we have seen in the previous lecture. So, organic nitrogen means the protein molecule, for example. So, organic molecule will be degraded. So, you will see that organic nitrogen is decreasing. Then after the degradation, what is produced? Ammonia. So, ammonia concentration, ammonia nitrogen will be increasing, and then finally it is converted to nitrite and finally to nitrate. This is the highest oxidation state of nitrogen. So, ultimately it will be converted to, it is of course in the aerobic environment. Okay. So, what is necessary then? If we want to know what is happening, then we have to know how to determine it. Different forms of nitrogen we have to determine, okay. That is the main thing. Now, why nitrogen data? Otherwise also nitrogen data is very important to know. Say for example, high nitrate content often causes methemoglobinemia in infants, okay. This is a disease, okay. So, so there is some permissible limit. So, if it is beyond that, then it may cause this type of disease, okay, in infants. Now, in the biological treatment, the reproduction of microorganisms are required, okay. We have seen in all biological systems the microorganisms should grow, right. So, if they grow, then the waste should contain sufficient nitrogen for the organism, because for the growth we always need the nitrogen. Say for example, protein molecule contains nitrogen, okay. So, determination of ammonia and organic nitrogen here, it is very important, okay. It is always required to know. Now, discharge of ammonia nitrogen and its subsequent oxidation can seriously reduce the DO level, that I already told you in BOD, okay. Nitrogen analysis is important to assess the problem, okay. Oxygen DO level should not go beyond some level, okay. Then anaerobic condition will be generated, that is not wanted, okay. Then determination of nitrogen are required to control the degree of purification produced in biological treatment. You all know this is very interesting. Ammonia, you know, ammonia is very toxic. So, there is some permissible limit 0.2 milligram per liter, permissible limit actually 0.02 milligram per liter. So, it should not go beyond this value, and ammonium ion is not, you know, ammonia fertilizer, ammonium sulphate, okay. So, this is very much needed for plant growth. So, ammonium ion is not toxic, but ammonia is toxic. So, we have to know how much ammonia is present, and for that we need some method. So, method is very important. Analytical chemistry is very important for the environmental engineers, that I will tell you, okay. How to determine the, everywhere it is a nitrogen, but nitrogen in different forms. Say for example, here it is ammonia, okay. Now, how you can determine the ammonia nitrogen? I will explain a very simple and very useful method, that is Nessler's method. You know, you are very familiar with this equilibrium. I told you in my chemical equilibrium lectures that ammonium ion is in equilibrium with ammonia plus H plus. So, if you want to shift this equilibrium towards right, then what you have to do? You have to take out this H plus, La-Shackler principle, I already explained to you. So, how you can take out? You have to make the pH alkaline, okay. So, this is very important, that will be required here when you determine this ammonia nitrogen. Now, ammonia reacts, this is the property, ammonia reacts with Nessler's reagent to produce a brown color having lambda max 450 nanometers. You are familiar with this, because in some of my lectures I already told you that spectrophotometer, I explained spectrophotometer, I also explained the spectrum or spectra, okay. 4-nitrophenol reduction, when I discussed about 4-nitrophenol reduction, there you have seen some spectra, okay, and you have seen some peak, UV visible spectra, there is some peak absorption spectra. So, this is absorption peak, that is called, where it is the maximum means absorption, there then it is called lambda, it is the wavelength, and here the peak is here 450 nanometers. It is a brown color. Okay. So, ammonia reacts with the Nessler's reagent to form a brown color, which is having the lambda max at 450 nanometers. Now, what is Nessler's reagent? Nessler's reagent is nothing but this is the AgCl2, okay, and excess potassium iodide in presence of NaOH. So, basically it is an alkaline solution of K2HgI4 and this is the reaction how it is formed. Okay. So, basically AgCl2 and Ki in presence of alkali it is forming this one. Now, the reaction of NH3 with this is represented as this is the Nessler's reagent, that is the anionic part, and then ammonia in presence of alkali it forms this is the complex. Okay. This is a complex which has the brown colored color, okay, and it is a colloidal solution that is formed. Okay. This is the balanced equation. It is also known as Milan's base. Now, you can use this method as a colorimetric method, because some color is developed. So, if you use some definite concentration of ammonia nitrogen and you mix the you perform this reaction, then as the nitrogen concentration is changing, so color intensity will also be different and you can use it as a colorimetric method. Okay. It is the brown color, intensity of the brown color is directly proportional to the amount of NH3 present in the solution. So, you can use it as a colorimetric method. Now, direct Nesslerization of the sample may lead to serious error. Direct Nesslerization means you take the sample and you put it, you put the reagent, that is the Nessler's reagent, then you think that some brown color will be formed and you can use this as for ammonia nitrogen determination, but it is not like that. Why? Because in the sample there may be other things also, okay, and that may lead to serious error because of the presence of interfering substances. So, the direct method is not possible. So, ammonia must be separated from sample by distillation. This is very important, and here comes this one. Okay. Say for example, ammonia nitrogen. What is ammonia nitrogen? Ammonia nitrogen is some type of nitrogen which is present either as ammonium ion or as ammonia. Okay. That is called ammonia nitrogen. It can be present either as ammonium ion or as ammonia. Okay. Now, this equilibrium you have to consider. Why? Because if you want to distill out this ammonia as a gas, okay, then you have to make the solution alkaline. Why? If it is ammonium ion, it will be solubilized in water, okay, but you have to convert it to ammonia. How you can convert? You have to shift the equilibrium completely towards right side, then you have to remove this. How you can remove it? Just making alkaline. Okay. You have to make sufficiently alkaline and then you have to use the Geldas flask. This is some type of distillation flask, okay, and there what you do, you take the sample, definite volume of sample, then you can make the solution alkaline by using say some alkali, say potassium carbonate you can use, and then you have to heat it. Then what will happen? The ammonia will be produced and it will be steam distilled. There will be steam also coming out from the solution, it is water solution, so steam will be there. So, ammonia will be carried out with the steam and then it will be taken out, it will be condensed to another flask. Okay. So, another flask, and there either you can take as some sulfuric acid, say for example, some acid you can take, so that ammonia goes there and it is converted to ammonium sulphate. Then ammonia will not go out because ammonia is volatile. So, ammonia may go out also. So, you have to take proper care. Okay. So, then after that you can take, you have to be sure that whole from the solution whole ammonia has 100% has come out. Okay. Otherwise, your result will be wrong. So, this is the thing, before boiling the sample water, a buffer solution is added to maintain the pH in the range of 9.5, and the distillate is collected into an acid solution contained in a flask. After this, so whatever interfering ions was there were there in your sample, they are not coming with the distillate, means coming with the ammonia vapor. Okay. They are remaining there in the that flask where from you are distilling. Okay. So, interfering ions are not present with ammonia. Now, then Nestlerization step is followed in alkaline pH condition. I have already told you what is Nestler's reagent. Now, what you will do, say for example, you have taken say 200 milliliter water, sample water, then you have to distill up to such time that at least 100 milliliter should come, even more is better comes, and then you will be sure that okay, all ammonia has come there in the sulfuric acid. Now, after it is collected there, then two things you can do. One thing is you can take the sample from there, and then you have to say for example 100 milliliter you are taking, and then you have to put the Nestler's reagent there. Immediately it will be forming brown color because of this. I told you that because of formation of this one, and then what you will do, you and another test tube, another tube, you have to take 100 milliliter of the distilled water, and then you have to mix it with the same volume of the Nestler's reagent, and then you have to add some source of ammonia nitrogen from outside. What is that? That is nothing but ammonium chloride solution. So, a standard ammonium chloride solution you prepare, where say for example 1 milliliter of that solution is equivalent to 0.05 milligram of nitrogen, ammonia nitrogen. So, you have to add drop by drop in that blank solution where you have taken the distilled water, and then when you will see the color of your sample water is matching. In the sample water also you have added the Nestler's reagent. So, it has already become some brown color, and now you are adding in the blank where you have taken distilled water, slowly drop by drop the ammonium chloride from outside, from a burette say, and then after some drop addition you will see that the color is matching. Then you will know how much ammonium chloride you have added, then you know how much nitrogen, ammonia nitrogen is present there. Okay. And it is same means color is matching, means whatever is there in the sample, that is same amount is present in the distilled water where you have added ammonium chloride from outside, then it is the same concentration. That is why it is called colorimetric, color matching. Colorimetric means color matching. You can extend this to spectrophotometry also, because you know that lambda max is this one. So, you can prepare some standard solution of ammonium chloride, and then you can add the Nestler's reagent, and then the color will be developed. They will have different intensities according to the concentration of ammonium chloride. Then you have some sample solution, there also you add the same volume of this Nestler's reagent, and you can match with all the solutions, it is colorimetric, or otherwise you can make a calibration graph using the spectrophotometer, using the standard solutions. Then you match, see how much absorbance you are getting for your sample, then from the calibration curve. I have already told you what is calibration curve in that lecture, previous lecture, that nitrophenol reduction. Okay. So, you already know how to make the calibration curve in the spectrophotometer. Here also you can use the same principle, but the reactions are different. Okay. Reactions are different. Okay. So, this is a very good method, you can apply it. Okay, for ammonia nitrogen determination. Now, what is organic nitrogen? What is organic nitrogen? You can apply the same method, Nestler's method, for to determine the organic nitrogen also. Now, you have to know what is organic nitrogen. All nitrogen present in organic compound may be regarded as organic nitrogen. It may be amino acid, it may be amides, it may be amines, it may be nitro compounds, it may be proteins. Most of the organic compounds containing nitrogen are derivatives of ammonia. So, if I ask you that in protein what is the oxidation state of nitrogen? Okay. So, protein is a very complex molecule. So, what is the oxidation state of nitrogen there? What is the oxidation state of nitrogen in ammonia? -3. So, here also it is same. When nitrogen has an oxidation state of -3. So, if you make something to decompose the organic part of the molecule by oxidation, that by oxidation releases the nitrogen as ammonia. Okay. Now, here actually when you decompose the organic molecule, that is, say for example, amines or ammonia, or not ammonia, say for example protein molecules, you decompose it, it is producing, first step is that it is producing the ammonia. Okay. There it is not oxidation, but later stage you can see, to make the reaction faster, a catalyst, this is the decomposition. Okay. So, to make this, it is a very complex molecule. So, you are degrading it, you are decomposing it to ammonia. It is a very tough work, tough job. Okay. So, it takes time also. To make the reaction faster, a catalyst such as mercury 2 plus or copper sulphate is used. This is a catalyst. You have already learnt what is catalyst. It makes a reaction faster. Okay. And then it is explained using some typical example. This is the alanine, you know, this is the amino acid alanine, alpha amino acid, which is oxidized by sulfuric acid in presence of catalyst for sufficient time till all the organic matter is completely oxidized. Okay. It is the actually nitrogen is not the oxidation state is not changing for nitrogen, but for carbon you can see that carbon is going to be carbon dioxide. Sulfur of that where from it is coming, sulfur dioxide is coming from sulfuric acid. So, you can see here, this is very nice change you can see. Once the organic nitrogen is released as ammonia and nitrogen, it can be measured in a similar way to that followed by the determination of ammonia and nitrogen by Nessler's method. So, distillation of the in the jet large flask you have to do, but then here sulfuric acid, concentrated sulfuric acid you have to add to the solution, okay, where you have some organic nitrogen present. Then you have to do it for sufficient time, you have to heat it at high temperature, you can see 360 degrees centigrade, okay, till the organic matter is completely oxidized. Okay. How do you know this? You will see lot of fumes are coming out, okay, because of this sulfuric acid, lot of fumes are coming out. First stage you will see that it has become black because of the formation of carbon. Okay. It is decomposed, so carbon is formed and then carbon dioxide will be formed. Ultimately, you will see that the black color has gone, it has become transparent solution, then you will fumes are also gone. So, in that stage you can tell that okay, the complete decomposition has occurred, and then this is the products that these are the products that is present. Okay. So, you can see here from complex molecule it has come like this. Now, it is ammonia nitrogen. So, previously I explained the how to determine the ammonia nitrogen. So, you have to follow the same process by using the Nessler's method. First you have to distill it in jet large flask, then you have to collect it in sulfuric acid, and then you can take some aliquot, definite volume of the aliquot, and then you can you can add the Nessler's reagent to get the color. So, the same way. Okay. Now, how we can determine the nitrate nitrogen, that is also Nessler's method, but here you have understood that in the Nessler's method we need ammonia, okay, or ammonia nitrogen. So, nitrate nitrogen is not ammonia nitrogen, it is present as nitrate or even nitrite. Okay. It is nitrite also, okay, nitrate nitrogen or nitrite nitrogen. So, how will you convert it to ammonia nitrogen? By reduction, simple. Okay. So, how will you reduce it? Determination of nitrate nitrogen or nitrite nitrogen is one of the most difficult tasks. It is very difficult for the analytical chemists or environmental engineers. So, nitrates and nitrites present in water can be reduced to ammonia by nascent hydrogen. You know nascent hydrogen, right? Nascent are very active reducing agent. How will you do it? The reduction of nitrate by nascent hydrogen can be done in different ways. This is one example. It is by Devar-Dash alloy. What is Devar-Dash alloy? Here you see alloy, you know there are many metals. So, copper is there, aluminum is there, zinc is there, and strong alkaline condition, alkaline solution. So, if you use this Devar-Dash alloy in strongly alkaline medium, then here it will produce the nascent hydrogen and then it will reduce nitrate to ammonia or nitrite to ammonia. So, now you are getting the ammonia nitrogen and now you can apply the same method. Another reducing agent may be zinc copper. This zinc copper, this is also some type of alloy. You can prepare it by preparing zinc by putting zinc granules to copper sulphate solution. You will see immediately copper is the blue color copper is deposited on zinc. Then after keeping from some time in acid condition, if you use, then it will produce the nascent hydrogen and then you have to follow the same method. After the reduction, then you have to follow the same method as you have done for ammonia nitrogen determination. Now, say for example, you have in your sample all 3 different types of nitrogen, but you want to know how much is ammonia nitrogen, how much is organic nitrogen and how much is nitrate nitrogen. Then you have to do it in stepwise fashion. First you have to take the same sample, you determine the ammonia nitrogen. So, ammonia nitrogen is gone there. After that you take the same sample from the gel dust flask, then you do the organic nitrogen and finally you do the ammonia that nitrate nitrogen. So, stepwise you can do, because if you just want to do the nitrate nitrogen first, then ammonia nitrogen will also come there. That is why it will not be correct. Okay. You have to do stepwise fashion, but method is same, but only thing initially you have to convert it, okay, to ammonia nitrogen. There are other methods also. Say for example, some spectrophotometric method. This is most simple method by using the spectrophotometer, by using the diazotization method. What is diazotization? Diazotization, diazo compound, N double bond N, diazo compound, it has very strong color. In textile dyes you will see that 90% dyes are having the diazo group, N double bond N group, they are having very intense color, okay. And then what you will do actually, there are 2 steps. In the step 1 is the sulfanylamide you will take and then nitrous acid, which is also nitrous acid means HNO2, okay. So, it is nitrite, okay. It is in acid form because you are using acid. So, it will ultimately it will be nitrous acid, but it is a method for nitrite determination. So, nitrous acid then they will react to form the diazonium salt, and then this diazonium salt will react with this compound N1-naphthyl ethylene diamine dihydrochloride to form a red colored azo dye. So, diazo compound to azo dye which is colored, okay. So, ultimately this is the colored compound that will be formed, and this you can measure the absorbance you can measure in the spectrophotometer. So, basically it is a very quantitative method, you can use it. The same method can be applied for nitrate analysis or total as nitrite plus nitrate nitrogen after cadmium reduction. So, this is basically a method for nitrite determination, but if you have nitrate, then you can convert the nitrate to nitrite first by some reduction. This is cadmium reduction, cadmium amalgam actually used, and then finally it is the nitrate plus nitrite you are getting total. If there is any nitrite, then you will get total, but if there is no nitrite, then you will get the nitrate. So, basically it is the same method, but another step is there, that is the reduction step. This method, this is ultraviolet spectrophotometric method for nitrate. this is a UV range, okay, 220 nanometer. This range is not very good, but some people use it because it is the most simple method, but you have to have this type of spectrophotometer. Ultraviolet range also should be there for determination, that is UV visible spectrophotometer. It is costly. It is much costlier than the visible spectrophotometer, but not that costly. So, we can use it, no problem. Then nitrate electrode is also there. This is the very simple method. You have electrode, you can just put it in the solution and you can get the concentration, but the shelf life of this electrode is not much, maybe 1 year or 2 year maximum, and it is costly also. So, but it is very user friendly, you do not have to do anything, only calibration and measurement, that is it. So, these are also applicable. Now, as a reference I can tell you these two books. One is the Sauer-McCarthy book that many times you have seen as a reference, and then A.K. Das, Asim K. Das, this is environmental chemistry with green chemistry, this is the publisher. This book is also very good, you can consult this book. Now, conclusion. In this lecture, what I have shown that nitrogen exists in water in 4 different forms, which are of interest to the environmental engineers. Estimation of ammonia nitrogen, organic nitrogen, nitrite nitrogen and nitrate nitrogen are discussed. So, this is basically analytical chemistry part, to know how much nitrogen, what is the different types of nitrogen present in the sample water. Thank you.